Welcome to Cell Biology. Today, we are going to explore one of the most fascinating and revolutionary events in the history of life on Earth, the endosymbiotic hypothesis. We will discover how a simple act of one cell engulfing another billions of years ago paved the way for all complex life, including humans. Let us dive in and see how our cells got their powerhouses. To understand how this biological merger happened, let us start with a simple analogy. Imagine a large, wealthy tech company that is struggling to develop a highly efficient battery. Instead of trying to invent it from scratch, they simply buy a tiny, brilliant startup that has already perfected the technology. Rather than dismantling the startup, the large company lets it operate independently inside its own headquarters. The startup gets safety and resources, and the large company gets all the amazing batteries. This is exactly what happened billions of years ago in the biological world, leading to the creation of complex life. Now that we have our analogy, let us look at the actual biological event. The word endosymbiosis comes from Greek, where endo means inside and symbiosis means living together in a mutually beneficial relationship. According to this widely accepted theory, around 1.5 billion years ago, an ancestral eukaryotic cell that did not use oxygen engulfed a free-living bacterium that could use oxygen to make energy. Instead of digesting it, they formed a permanent partnership. Over millions of years, this engulfed bacterium lost its ability to live independently and became a specialized organelle known as the mitochondrion. A similar event happened later when photosynthetic bacteria were engulfed, eventually becoming the chloroplasts found in plant cells today. This incredible ancient merger sounds like science fiction. So how do we know it actually happened? The evidence is overwhelming and can be found right inside your cells today. Mitochondria do not just rely on the host cell. They have their own circular DNA, much like bacteria. They also have their own ribosomes to build proteins, and they divide completely independently of the host cell's division cycle. These characteristics are exactly what we see in modern bacteria, serving as a microscopic fossil record that proves mitochondria were once free-living organisms before they were engulfed. Understanding this evidence leads us to ask why this event is so crucial to biology. Simply put, without endosymbiosis, you and I would not exist. Before this event, life was limited to simple, single-celled organisms that extracted very little energy from their environment. Acquiring mitochondria gave cells a massive energy source, allowing them to grow larger and evolve into animals and plants. Furthermore, in modern medicine, knowing that mitochondria are ancient bacteria helps us design antibiotics safely, avoiding drugs that might accidentally attack our own cells. It also allows scientists to map human migration by tracing mitochondrial DNA passed exclusively from mothers to children. Before we move on to the mathematics of this energy leap, let us visualize this process. On the screen, you can see a diagram illustrating the endosymbiotic event. On the left is the large ancestral eukaryotic cell, which already has a nucleus. It is in the process of engulfing the small, red, rod-shaped aerobic bacterium. Following the arrow to the right, we see the final eukaryotic host cell. Notice the mitochondrion now has two membranes. The inner membrane comes from the original bacterium, while the outer membrane was formed by the host cell during the engulfment process. This double membrane is a key physical proof of the hypothesis. Now that we have seen how the cell acquired the mitochondrion, Let us look at the massive energy advantage it gained mathematically. The universal energy currency of the cell is a tiny molecule called ATP. We can represent the total energy production using a simple equation. E total represents the total amount of energy produced from one molecule of glucose, measured in ATP. E glycolysis represents the tiny amount of energy produced by the host cell's original method, which does not use oxygen. E oxidative represents the massive amount of energy produced by the newly acquired mitochondrion using oxygen. When E oxidative increases from 0, the total energy skyrockets. Let us apply this formula to a concrete example to see the exact energy advantage gained. We are given that the ancient anaerobic cell can only perform glycolysis, producing exactly 2 molecules of ATP per molecule of glucose. After engulfing the bacterium, the cell can now perform oxidative phosphorylation, which adds an additional 34 molecules of ATP. We want to find the new total ATP yield. We substitute 2 for E glycolysis and 34 for E oxidative into our equation. Adding these together gives us a final answer of 36 ATP molecules. This massive pool of energy allows the cell to perform complex tasks. Having calculated the new total energy, we must now understand how much of an upgrade this truly was. To do this, we calculate the fold increase, which we will call F. This formula simply divides the new total energy, E total, by the old total energy, E glycolysis. The fold increase tells us exactly how many times more powerful the cell became after the endosymbiotic event. If the total energy increases while the original glycolysis energy remains the same, the fold increase value goes up. This mathematical ratio is the key to understanding why complex life was finally able to flourish on Earth. Let us use our previous results to calculate this exact power upgrade. We know the new total energy capacity of the cell is 36 ATP. We also know the original energy capacity was only 2 ATP. We are asked to find the fold increase in energy production. By dividing 36 by 2, we get a final answer of 18. This means that by simply engulfing this tiny bacterium, the host cell instantly became 18 times more efficient at extracting energy from its food. This monumental 18-fold leap in biological efficiency is the physical reason complex life evolved. This incredible energy advantage came with a trade-off, which brings us to the mathematics of genome reduction. Over billions of years, the engulfed bacterium transferred most of its DNA to the host cell's nucleus. We can calculate the absolute number of genes lost, which we will call L, using a simple subtraction formula. G original represents the starting number of genes in the ancient free-living bacterium. G current represents the number of genes remaining in the mitochondrion today. As the bacterium becomes more dependent on the host, G current decreases, meaning the number of genes lost increases over evolutionary time. Let us see exactly how many genes were transferred by looking at a real example. Suppose the original free-living bacterium had a genome consisting of 3,000 genes. Today, a typical human mitochondrion retains only 37 genes in its own circular DNA. We need to find the absolute number of genes that are no longer present in the mitochondrion. We subtract the current 37 genes from the original 3,000 genes. Our final answer is 2,963 genes. These genes have been safely transferred into the DNA vault of the host cell's nucleus.
Now that we know the absolute number of genes lost, we can express this as a percentage to truly grasp the scale of this reduction. The formula for percentage lost, P, takes the absolute number of genes lost, L, and divides it by the original number of genes, G original. This gives us a decimal fraction. We then multiply that fraction by 100 to convert it into a readable percentage. If the number of genes lost is very close to the original number of genes, this percentage will approach 100, indicating a near total loss of genetic independence.
Let us finalize our genome reduction calculation by finding this percentage. We know from our previous step that 2,963 genes were lost. We also know the original genome had 3,000 genes. We are asked to calculate the percentage of the original bacterial genome that has been lost. We divide 2,963 by 3,000, which gives a decimal. Multiplying by 100 gives a final answer of approximately 98.77%. This physically means the mitochondrion has surrendered nearly 99% of its genetic independence. We have covered a lot of ground today, so let us review the most important ideas. First, the endosymbiotic hypothesis is the ultimate story of biological teamwork, where complex cells evolved by engulfing specialized bacteria. Second, this permanent partnership provided a massive 18-fold energy advantage that fueled the evolution of all complex life. Finally, while the mitochondrion lost nearly 99% of its original genes to the host, its double membrane and circular DNA remain as a microscopic fossil record. You now understand the fundamental event that bridges simple bacterial life and the complex cells in your body.